Hey guys, welcome to the lecture on Chapter 5, which is all about carbohydrates. Carbohydrates are another class of biomolecules. If you remember last chapter, in Chapter 4, when we started talking about organic chemistry, the organic molecules that are most important in biological systems are the biological molecules, which are carbohydrates, fats or lipids, proteins, and nucleic acids, so DNA and RNA. So we've already briefly talked about fatty acids and lipids. We talked about those in Chapter 4. Chapter 5 brings us to carbohydrates. These biomolecules may sound familiar to you because they are part of our food. They're nutrients. They're actually molecules that we get from food, but a lot of them are molecules that we synthesize as well. Or more importantly, they are molecules that we get from food and that we then break down either for energy or to make and synthesize chemicals that are important in our body. And Chapter 12, when we get to that at the end of the semester, is all about metabolism, how we break down these large biomolecules and use them for energy. But before we can learn about how we break them down, first we need to learn about what they are and what they look like and what their properties are. So in this chapter, we're going to talk about the different classes of carbohydrates, which include monosaccharides, disaccharides, and polysaccharides. We're also going to talk about the structure of carbohydrates. A lot of times they're written in like a linear structure that we're going to introduce them as, but they actually form rings. So we're going to talk about the chemistry of how those rings actually form. And we're also going to get a little introduction into a chemical reaction, a type of chemical reaction called oxidation and reduction, or also known as redox reactions. These are very popular chemical reactions that take place in living systems, particularly in metabolism. So we're going to learn about the basic chemistry of oxidation and reduction, and then again in Chapter 12 we'll come back to it and see it again when we talk about metabolism. So what are carbohydrates? Carbohydrates are organic molecules, like I said. They are biomolecules, and they can be divided into two classes, into sugars, which are simple carbohydrates, and polysaccharides, which are the complex carbohydrates. Okay? Carbohydrates are mainly important. We get them from our diet, and they're mainly important for giving us energy. We break them down to make ATP. When we talk about energy, we're talking about making ATP for our cells. So that's the major role of carbohydrates, though we will talk about some other roles of carbohydrates in our cells and our body at the end of the lecture. So where do carbohydrates come from? Carbohydrates are the products of photosynthesis, that process where plants take carbon dioxide from the air and light energy from the sun and water from the earth and from rain, and they combine them all together to make glucose, a carbohydrate called glucose. So here you go. This is where carbohydrate, the term actually comes from. It's from carbo, as in carbon dioxide, hydrate, as in water. You mix the two together with a little energy from the sun, and you get a carbohydrate. So this C6H12O6 is the chemical formula for glucose. It's a simple sugar, a simple carbohydrate, and it is the most abundant carbohydrate in nature, the most abundant carbohydrate in our bodies, the most important carbohydrate probably on the planet. And as a byproduct of this chemical reaction, photosynthesis, plants also produce oxygen, which is why they're important for us to breathe, and why we're important for them to breathe, I suppose. So like I said, there's different classes of carbohydrates. So you have your simple sugars, which are going to be your monosaccharides and your disaccharides. And then a couple slides I'll talk about the polysaccharides, the more complex carbohydrates. So these simple carbohydrates, monosaccharides and disaccharides, are simple molecules. A monosaccharide is a single sugar ring, whereas a disaccharide is two sugar rings bonded together. Di meaning two. So glucose is a monosaccharide, the most abundant monosaccharide. Other monosaccharides include fructose, which is very sweet, which is why we have come up with high fructose corn syrup. It has a little more fructose than it does glucose, and that's why it's a little bit sweeter. Sucrose is an example of a disaccharide, and sucrose is table sugar. It's the stuff that you use to sweeten your coffee, and it's sweet because it's a combination of glucose and fructose, fructose being, again, the sweetest of the monosaccharides. So once we start adding monosaccharides to our chain and making it a little bit longer, we call these longer chains of sugars oligosaccharides and polysaccharides. Oligo means few, so an oligosaccharide is 3 to 9 monosaccharides joined together, and a polysaccharide is anything that's 10 or longer. And we don't really have any oligosaccharides in our diet. We'll talk about the role of oligosaccharides when we talk about blood group antigens, A, B, and O blood group antigens. But polysaccharides are something that are very prominent in our diet, because what happens is plants make all this glucose, but plants don't really need to use a lot of glucose. I mean, what are they doing? They sit there and they grow leaves and flowers, but they don't have to run around, they don't have a lot of organs that have to function. So a lot of that glucose that they're making ends up going into storage, and they're not going to store a bunch of free glucose. What they do is they link all those glucose molecules together in these long polysaccharide chains in order to store it for later, in case the plant needs energy later, like during the winter months or something where it can't photosynthesize, but also for the benefit of other animals higher up in the food chain that eat those plants. Okay, so polysaccharides, there's a couple of forms of them. We're going to talk about the two different forms of polysaccharides being starches and fiber. And actually there's a third form called glycogen, which is in animals, and we'll talk about that too. But polysaccharides are just long strings of monosaccharides, poly meaning many. So when it comes to sugars and carbohydrates, we've got a couple of new functional groups we're going to be exploring. So remember in Chapter 4, Table 4.6, I think it was, there was a list of all the different functional groups, okay? And we didn't really talk about all of them, but hopefully you did the homework and you started practicing being able to identify different functional groups. But the ones that we're going to explore in this chapter include alcohol functional group, the aldehyde functional group, and the ketone functional group. So I'll get to those in a minute, but first I want to mention this, that all monosaccharides have a general formula of CN H2O okay? So if you look at the photosynthesis process and look at glucose, you'll see why that is. So if you recall, carbohydrates are made, monosaccharides are made by taking carbon dioxide and water. Okay, so 1 carbon plus 1 water equals, so I guess actually this is not a balanced equation. It should be 6 carbon dioxides and 6 waters become C6H12O6 with a little bit of excess oxygen. Okay, so let's just do a little practice here. See if I can do this. Can't draw in Photoshop, in a PowerPoint for some reason, so I have to draw in Photoshop. Okay, so let's say our N is, we have a carbohydrate that has 8 carbons. So what would the formula for that be? Our N is 8, so it would be C8 H, and we remove the parentheses, so it would be H, so it would be 2 times N, 2 times 8 is 16, and O to the N would be 8. So there's a pattern. So this would be a carbohydrate, C8H16O8, okay? So similarly, I could give you a problem that says, is C8H16O8 a monosaccharide? Is that a general formula for a monosaccharide? And you could work backwards and say, yes, it is. Essentially, it should be the same number of carbons and oxygens and twice as many hydrogens, okay? Is the general formula for a monosaccharide. Alrighty, so back to the PowerPoint here. So let's first talk about the alcohol functional group. We're going to talk about all these different functional groups now. The alcohol, aldehyde, and ketone functional groups. So when we talk about alcohol functional group, yes, we are talking about the alcohol in alcoholic beverages. Ethanol is the alcohol in alcoholic beverages, and it looks like this. The formula is CH3, CH2OH. It's an ethane molecule with an alcohol group. That's how it gets its name, ethanol. Okay, so all alcohols have this suffix "all," and they all have this functional group, this OH, okay, or a hydroxyl group. Because remember, hydroxide is the OH minus ion. It's a polyatomic ion, OH minus, hydroxide. So this is the hydroxyl group. So alcohols can be classified based on how many carbons that alcohol functional group, well, I should put it differently. The carbon that the alcohol functional group is bonded to, so in this case this second carbon here, all right, is referred to as the alcoholic carbon because that's the one that the alcohol is bonded to. So alcohols can be classified based on how many other R groups this alcoholic carbon is bonded to. So you can have primary, secondary, and tertiary alcohols. So in this case, let's do a little drawing here. Okay, so in this first case here, we have a carbon, the alcoholic carbon here, because it's bonded to the alcohol group, is bonded to two hydrogens and a methyl group. So anything that's not a hydrogen and not a functional group, we call an R group. R means the rest of the molecule, okay? So this carbon is bonded to one R and two H's. This carbon is bonded to two R's, so this methyl group is an R, and this one is an R, and an H, and this alcoholic carbon is bonded to three R groups. I hope everyone's able to understand this. Okay, so if it has one R group, it's a primary alcohol. If it has two R groups, it's a secondary alcohol. If it has three R groups, it's tertiary alcohol. All right. This is demonstrated really well in that table from Chapter 4, Table 4.6. Okay, and here it labels the R groups for you. So R could be a methyl, like a CH3. It could be CH2, 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 CH3. It could be a butyl group, a propyl group. Okay, it could be any it could be any hydrocarbon group out there. So these are the primary, secondary, and tertiary alcohols. This is a molecule of glucose, okay, which has, and now you can see the alcohol groups. Here's the aldehyde group we're going to talk about in a minute. But there's 1, 2, 3, 4, 5 alcohol groups, functional groups, in a glucose molecule. 4 of them are secondary alcohols because, let's see if I can highlight this. Okay, so for this carbon right here, all right, the second carbon from the top, here's R group number 1. This whole thing up here is a group, okay? Then there's a hydrogen here, and then there's a whole group down here. All right, so we've got R, and then a hydrogen, and the alcohol functional group. So this carbon has two R groups bound to it, which makes it a secondary alcohol, okay, as it says here. This last one here, the alcoholic carbon, has two hydrogens and one R group, which is this whole thing up here, okay? Two hydrogens, one R group, let's look at the table here, is a primary alcohol, okay? So there's several practice problems like that in the chapter about identifying primary, secondary, tertiary carbons or alcohols, and we'll do one right here. All right, so this molecule here is it primary, secondary, or tertiary? So another way to show this molecule, CH3, sometimes it's a little hard to I think it's hard to visualize in condensed structure, so sometimes I like to draw it out a little bit more just for my own sake. Okay, so CH2, CH2, and then I'm going to draw, this is the alcoholic carbon, okay, there's three things attached to it. All right, two are hydrogens, CH2, so we'll draw a hydrogen here and a hydrogen here. So this is kind of a sort of condensed Lewis structure here. And then there's also an alcohol group that's attached to it. So here's our oxygen and our hydrogen. Okay, so looking at this molecule, here's the alcohol group, here's the alcoholic carbon, there's two hydrogens and one R group. Okay, so this would be what? This would be a primary alcohol, because it only has, this alcoholic carbon only has one R group. Okay, and how about this one? We've got, I'm going to draw in here, the ends of the lines are carbons, right? So these are CH3 groups at the end, and sometimes that's hard to see, so I'm just going to write it in here that this is a CH3 at the end of this skeletal structure, and this is a CH3. Sorry, it's not very good at drawing with my trackpad. CH3, okay? This over here is a CH3 too, but I'm not concerned about it, so I'm not going to draw it. And right here, this intersection here, this is our alcoholic carbon, okay? This is the one that's bonded to the alcohol group. So how many R groups does this have? It has 1, 2, 3 R groups, okay? So 3 R groups means 3 prime, so tertiary. It is a tertiary alcohol, okay? So hopefully everyone sees that. All right, moving on to aldehydes and ketones, another type of functional group. And both aldehydes and ketones also are functional groups that contain carbon and oxygen, okay? But that are bonded in a very special way. So both groups contain this type of a bond called a carbonyl. Okay, that's how you pronounce that, carbonyl. It's a carbon double bonded to an oxygen. So an aldehyde is a carbon that's double bonded to an oxygen and then has a hydrogen on one side and usually has an R group on the other side. Think of this other hydrogen here as an R group. Formaldehyde is the only exception where it has two hydrogens bound to it, so it's the simplest aldehyde. Usually an aldehyde is just this. It's a carbon, it's a carbonyl that's with the carbon is bonded to a hydrogen, and then this can be an R group over here. And a ketone is a carbonyl that's bonded to two R groups. So aldehyde, carbonyl bonded to one R group and a hydrogen, and ketone is a carbonyl bonded to two R groups. Okay? I thought I had the table. Anyway, table 4.6 in your book has a good example of them too. So, looking at the structures of some monosaccharides, you see that some of them have an aldehyde group and some of them have a ketone group. So sugars, the suffix for a sugar is -ose. Lactose, glucose, sucrose. All right, notice they all have that same suffix, -ose. So if a sugar has an aldehyde in it, we call it an aldose. And if it has a ketone group, we call it a ketose. Simple as that. So let's practice identifying aldehydes and ketones, okay? Slide 13. All right. This one here. So the first thing we want to do wants to know, is it an aldehyde or a ketone? If we're looking for aldehydes and ketones, the first thing you want to look for, you want to zone in on that carbonyl, okay? So I'm going to go ahead and circle the carbonyls here, or attempt to, okay? So here's the carbonyl in this molecule. So this carbonyl, this carbon is bonded to one hydrogen and one R group. This whole thing here is an R group, okay? So it's got one R and one H. So does that make it an aldehyde or a ketone? The correct answer is aldehyde, okay? So hopefully you got that. Aldehyde. How about this molecule? We have our carbonyl here, all right? And then we have a CH3, right? The end of this line here represents a CH3, a methyl group. So I'm going to draw that in for clarification. I do that partly for you, but partly for me, because I get easily confused sometimes when things aren't explicitly written out. So I'm very visual, and I encourage you guys to give yourselves visual clues as well when problem solving. Okay, so this carbonyl is bound to an R group on this side and an R group on this side. So does that make it an aldehyde or a ketone? The correct answer is it's a ketone. If it's got two R groups, it's a ketone. The last one here we go. Here's our carbonyl, and it's bonded to an R group on the left and a hydrogen on the right. So one R group means that it's an aldehyde, just like the first one. All right? Good job there. Practice more of those. All right, so when it comes to monosaccharides, biomolecules in general, are larger molecules. I mean, they're not just, they have chains of carbons linked together. They're a little bit larger than molecules when we've talked about binary compounds and things like that. And when it comes to the stereochemistry, which is a fancy word for talking about isomers, okay, stereoisomers, monosaccharides have several chiral carbons, meaning they can have several stereoisomers, several enantiomers, okay, or several molecules that have the same molecular formula, same connectivity, but they are mirror images. So we have a molecule of glucose here, and if we count the chiral carbons, they're already indicated in this picture. Carbons 2, 3, 4, and 5 are all chiral carbons. They are all carbons that have 4 different groups attached to them. Okay, so if there are 4 different chiral carbons, that's 4 places where there can be a mirror image of that carbon. So there can be several stereoisomers of glucose. The formula to calculate how many stereoisomers of a molecule you can have if it has multiple chiral carbons is 2 to the n, where n is the number of chiral carbons. So in this case, glucose has 2 to the 4, which means it has 16 different stereoisomers. Okay? So that's kind of amazing if you think about the fact that there are 16 different stereoisomers of glucose, yet the one that our bodies have in most organisms, living organisms on our planet, have adapted to use primarily as a source of energy is D-glucose, this one specific stereoisomer. All the other stereoisomers are used for other things, or they're not processed or recognized at all. So it's kind of crazy to think that of 16 different possibilities of one molecule, we've picked one that we use. And glucose is not the only biomolecule out there that has multiple different stereoisomers. All of the molecules that we use in our body, we prefer a specific isomer, a specific stereoisomer. It's all about that chiral recognition. The enzymes in our bodies and receptors in our bodies recognize specific forms of molecules. So it's kind of crazy to think about how many molecules are out there that our bodies don't recognize or utilize also. So chemistry is crazy. All right, so there's a little bit of technicalities in this chapter talking about how we draw molecules, different shortcut ways to draw molecules to simplify things. So one way is to use these wedge and dash drawings. The wedges are the dark colored filled in ones, and they indicate that something is coming out of the page at you, whereas these little dashed markers, okay, are supposed to indicate that something's going back into the page, behind the page. It's a way to indicate the three-dimensional structure. But a quicker way to draw these molecules is to do Fischer projections, okay, where we just use lines, vertical and horizontal lines, to draw a molecule, and the intersection of those lines indicate the chiral carbons. So we don't necessarily draw the chiral carbon, as you see here. It's just assumed to be at that intersection, those crossroads. So then we don't have to bother with coloring in dashes and making dashed lines. Okay, it's much quicker to draw them this way. And you'll see that when we do some exercises. So here's a Fischer projection. So these line molecules here are called Fischer projections, named after some guy named Fischer who invented them. So Fischer projections make it really easy to draw enantiomers of our monosaccharides. So remember, D-glucose is the isomer, the enantiomer of glucose that we primarily use, well, that we exclusively use. We don't process L-glucose. We can only process, our enzymes in our body only recognize D-glucose. So this is a molecule of D-glucose. D means dextero. It indicates the right. So remember, with enantiomers, you have right-handed ones and left-handed ones. So the right, I just did that wrong, right-handed and left-handed. So D and L is another way to say right and left. Also, R and S is another indication of right and left isomers, and plus and minus. So there's different, I don't know who coined all these different terms and why it's not, hasn't been unified, but D and L are the ones you use the most when it comes to biochemistry. So D and L, D for dextero, right, and L for left, or levitus or something is the Latin term. So the reason we get this right-handed, left-handed, it happens to do with the last alcohol group on the last chiral carbon, okay, whether it's on the right side of the Fischer projection or the left. So it's on the right, then it's D-glucose, right-handed glucose. If it's on the left, then it's L-glucose, left-handed glucose. So that's just where that nomenclature comes from. So let's draw some Fischer projection enantiomer mirror images of these guys. Okay, so remember, only chiral carbons have mirror images. We only need to draw mirror images of the chiral carbons. These carbons up here, the one at the top and the one at the bottom, the ones that are not chiral, we don't have to make them mirror images. Okay, so you can just draw them as is. I'll draw the group here. Here's my carbonyl, starting with it. Now I'm adding an H, which makes it an aldehyde. All right, and then let's draw out a long line. The 6th carbon down here, CH2, OH. You should be right. Anything I write, you should be writing in your notes. I probably should have mentioned that day one, but it's never too late. So if I'm writing it, you should be writing it and practicing it with me, not just following along. Plus, this makes me go really slowly, so you should have plenty of time to write with me. Okay, so we're going to do the mirror image. So in this first carbon, H is on the left, OH is on the right. We're going to flip it. We're going to put OH on the left, OH. I went over the mirror a little bit here, and now H is on the right. All right, this one, flipping them around. So now H is on the left instead of on the right, and OH is over here, and so on and so forth. So you'll see that all of the groups are facing each other in the mirror. So the OHs are now facing, H's are facing each other. So that's another way to do it, is to draw the groups facing each other, like they would in a mirror, and so on and so forth. And this one's going to be an OH. I'm going to draw it as an HO. Ta-da! So now we did D-galactose. We just drew L-galactose, left-handed galactose. I'm just going to write L-gal. All right, that's what we would call this. Let's do one more. So we draw out our non-chiral carbons. We don't, I mean, you can draw them as a mirror image. It's not wrong to draw it this way. I'll draw it this way. Why not? Carbon. I'm drawing the mirror image here, but it doesn't matter. All right, this looks really weird. You would actually never write it this way. It should always be CH2OH. Okay. 1, 2, 3 chiral carbons. So this one, we'll do mirror image, H, so on and so forth. Hopefully you've already finished drawing this one. All of the hydrogens are going to be on this side, and all of the hydroxyl groups, alcohol groups, are on this side, the left side. So now they'll all be facing each other. Okay, ta-da! So now we have L-ribose. L-ribose, left-handed ribose. L-rib. Okay. Whew! A lot of work trying to draw with this trackpad. Okay, so to make things a little more complicated and add a little more vocabulary, so in Chapter 4 we talked about isomers. So we talked about structural isomers and conformational isomers. Those are isomers of connectivity, where there's differences in the connectivity. And then we talked about the stereoisomers, which included cis and trans isomers, and also the enantiomers. Okay, so to complicate things a little bit, when it comes to enantiomers and stereoisomers, there's also some other, especially when it comes to molecules that have multiple chiral carbons, you can have a full enantiomer. Okay, so that's what we were doing here. We were drawing the enantiomers, so the full-on mirror image of that molecule. But when you have multiple chiral carbons, you can have enantiomers at some chiral carbons, but not at others. So when you don't have the whole molecule is not a mirror image, only certain chiral carbons are mirror images, we call those diastereomers. Okay, diastereomers. And if a diastereomer differs in only one carbon, one chiral carbon, then we call it an epimer. Okay, so we've got 3 words here that need distinguishing. Enantiomers, which are full-on mirror images, okay? Diastereomers, which would be like if I took my hands and I somehow flipped a couple of fingers or something. So diastereomers have a couple of groups flipped, but not the whole molecule. And an epimer just has one group flipped, okay? So here's some examples. You have D-glucose here, And if you compare D-glucose to D-galactose, you'll see that only the carbon on the third one here is flipped. So that would be an example of a diastereomer or an epimer. That would be an epimer because only one chiral carbon is flipped. And D-talose, these two blue groups here, those are the two that differ, that are flipped from D-glucose. So D-talose would be a diastereomer of D-glucose, okay? But notice in red here, they've highlighted the fifth chiral carbon to show you that all of them have the alcohol group on the right side. So they're all still D molecules. They all still D-glucose, D-galactose, D-talose, all right? Whereas if it was a full enantiomer, it would be the L version, okay? So they're all still right-handed, but they're all diastereomers of glucose. So here we'll do a problem where we are indicating whether the molecule is an enantiomer, a diastereomer, or an epimer, okay? So here's D-mannose. I need to do my little copying and pasting job. All right, so we have D-mannose, and we want to determine whether A, B, and C are enantiomers, diastereomers, or epimers. So let's look here. In A, is this first chiral carbon flipped? Nope, it's the same. OH on the left, hydrogen on the right. Yep, this one's the same. How about this one? Oh, nope, this one is flipped because the OHs, the alcohol groups, are facing each other here in the mirror. They're facing each other, all right? And this one is not flipped. So this one only has one group flipped. What do we call that? It's a specific type of diastereomer called an epimer. I'm just going to write epi for short, okay? An epimer. All right, now let's compare D-mannose to this molecule B. Let's see, this first group is flipped here. This second group is flipped here. The hydrogens are facing each other. The third group is flipped. And it looks like the fourth group is flipped as well. So all of the chiral carbons are mirror images of each other. Do you see that? So then this one would be called what? An enantiomer. Enantiomer. Okay? And now for molecule C. Let's see, the first one, nope. Second one, yes, you are mirror images. The third one, yes, mirror image. And the fourth one, no. So two groups are flipped. That makes this a diastereomer. Diastereomer. Okay? So practice those. This is just a matter of remembering the different names for each. And keep in mind that for doing homework and for this next test, the test that's going to cover Chapter 5, you will have open book. So if you just make it yourself a little cheat sheet, a little page or something that says with tips and notes to complete homework problems, epimer is one chiral carbon as a mirror image. Enantiomer, all chiral carbons are mirror image. And diastereomer, more than one, but not all. Chiral carbons are mirror images, okay? The good news is you don't have to keep it straight in your head as long as you keep it straight in your notes somewhere and can do the work through the problems that way. Okay, so there's a lot of important monosaccharides in our diet, in our bodies. Glucose is the most prominent in the world, really. Another name for glucose is dextrose, just FYI. And it's our blood sugar. So like 90% of the sugar in our body is in the form of glucose. And we break glucose down for energy through metabolism in a process called glycolysis, which literally means breakdown of glucose. Galactose is another monosaccharide in our diet. Galactose is a monosaccharide, and when you combine it and bond it to glucose, it makes lactose, milk sugar. So we don't really, there's no like galactose by itself as a monosaccharide in food. We see it after we break down lactose in our bodies. Mannose is another one that's actually not really broken down by the body, and it's found in a lot of fruits. It's actually one of the sugars, one type of sugar that can give you diarrhea if you eat too much of it, because your body doesn't absorb it, so it gets excreted with the feces, and it actually attracts water. But it's sort of claim to fame that they highlighted in the book is that bacteria, really, some of the bacteria on our bodies or in our bodies really like mannose, and they can break it down, particularly the bacteria that cause UTIs. So if you eat a lot of, and cranberries tend to have a lot of mannose, so eating a lot of cranberries provides all this mannose in your urine, and the bacteria will bind to it and then be excreted with the mannose. So it helps to sort of flush bacteria from the system, apparently. That's how it works. So fructose is I would say the third. I don't really know why they choose to talk about mannose, because it's not really an important monosaccharide in our daily life. But fructose is another one that's really important. It's found in table sugar, the added sugars to our foods. The most common sort of added sugar to our food, sucrose, is half glucose and half fructose. It's a disaccharide made up of fructose and glucose. So there's also free fructose in our food, in fruits. That's where it actually gets its name from, fruit sugar, fructose. And it is the sweetest of the monosaccharides, so it's what we really crave when we're craving sweetness. So all the dietary monosaccharides are hexoses, meaning they have 6 carbons, okay? So here you have fructose, glucose, and galactose. All of them have 6 carbons. You can count them. And we aren't going to talk about pentoses really in this chapter, but pentoses, having 5 sugars that have 5 carbons, are also biologically important, but not in our diet, not for energy, for DNA, for an RNA, for our nucleic acids. And unfortunately, we're not going to have time in the semester to go over the chapter on nucleic acids, but your DNA is your genetic material. It encodes all of your genes. And it's pretty important stuff. And RNA is sort of this molecule that makes photocopies of the DNA in order to send to the machinery that makes proteins, that turns that DNA into proteins. So here are the bases, the nucleic acid bases. Or here, I guess here's a nucleic acid base. ATP is that energy molecule that we use for energy. It also happens to be a base for the nucleic acids. But you can see here, this part here, this pink part, is a sugar ring. It's a ribose. Okay, ose being the suffix for sugar. It's a pentose. It's a 5-carbon sugar. And then there's adenine and this triphosphate. So that's what actually ATP stands for, adenosine triphosphate. So you have an adenosine, and then you have 3 phosphate molecules here. And when it comes to DNA and RNA, you have this 5-carbon deoxyribose that makes up your DNA, deoxyribose DNA, and RNA has ribose. Instead of deoxyribose, it has ribose here. So these are 5-carbon sugars, 1, 2, 3, 4, 5. They're pentoses. And they're very important for our nucleic acids, okay? But, we might maybe mention them again later in the semester. And if you have time and you're interested, it wouldn't hurt you, especially in preparing for some other classes that you might be taking entering allied health, like human bio or microbiology. Reading chapter 11 is one chapter we won't get to go over, but it's all about the nucleic acids. Okay, so those are all the important sugars. And now the chapter, we digress a little bit, I think. to talk about redox reactions, which redox is just short for reduction and oxidation. So redox reactions are chemical reactions that involve losing and gaining of electrons. It involves a transfer of electrons. It's different than when we're talking about transferring electrons between atoms to form bonds, because in that case you're so for an ionic bond, like sodium will transfer an electron to chlorine, and then those two ions are attracted together and they form a bond. All right, sometimes there's chemical reactions where molecules give electrons and another molecule takes the electrons and then they go their separate ways. They don't form a bond, okay? So that's what redox reactions are. So there's a little mnemonic to help remember which is which. So when atoms in a reaction lose electrons, okay, that is called oxidation. So oil rig means oxidation is loss, reduction is gain. Oil rig. Oxidation is loss, reduction is gain of electrons, okay? So some examples in chemistry, inorganic chemistry, of oxidation, of redox reactions are the patina that forms on copper when it's oxidized. So the Statue of Liberty is actually made out of copper. It's copper color. It should be the color of pennies, okay? But being out in the ocean, open, exposed to air and salt and water, all things that promote these redox reactions, it forms essentially a type of rust called patina, which is green colored. So that's why the Statue of Liberty is green. It's actually made out of copper, and this is essentially rust. Same thing happens to your pennies. I have a whole bunch of pennies in the cup holder in my car, and I'm always putting, my water bottle there, or like, my iced coffee in the summer, and so water drips onto the pennies, and they rust, and they form this green patina. They're really ugly pennies. Rust is what happens to iron. It's oxidation of iron, and it's an orange color. So it has to do with the chemical that's formed when iron gets oxidized versus when copper gets oxidized. When copper gets oxidized, it turns green. When iron gets oxidized, it turns like this reddish-brown color, and that's what rust is. And you get rust. You see this pattern of rust around a lot of cars, but particularly up here in the North Country. I never noticed this pattern before I moved here. And I was curious why that was, and I looked it up one time, and it's because all the grit and salt and pebbles and stuff on the road get kicked up by your tires and chip the paint around I don't know what you call this thing, the tire well or something like that, okay? So the paint gets chipped, and the paint is protective. The paint covers the metal, prevents it from oxidation, prevents the iron from rusting. But when you chip the paint, then you let oxygen in, and salt, particularly salt from the roads during the wintertime, actually catalyzes this reaction. So you get a lot of rusting here in North Country. So wash your cars in the wintertime. Try to keep the salt off. It'll slow the rusting of your car. So anyway, here's a nice little diagram. I like this diagram of redox reactions because there's a little, so they can be a little bit confusing because with this whole reducing agent, oxidizing agent, and what gets oxidized and what gets reduced. Again, it's like a tongue twister, okay? So you have two atoms here, two molecules. One is going to give up its electrons and the other is going to accept it. So remember with oil rig, oxidation is loss and reduction is gain. So this orange molecule here loses an electron. Oxidation is loss, that means it got oxidized, and reduction is gain. This guy gained an electron, so it got reduced, okay? And since one can't happen without the other, what that means is that the guy that's losing an electron is the reducing agent. This one who gets oxidized, the molecule that gets oxidized, acts as a reducing agent. It's an agent that reduces another molecule. Likewise, the molecule that accepts the electrons and gets reduced in the end is the oxidizing agent, okay? It's a little bit confusing, but if you think about it, it makes sense. The one that gets oxidized, because it's a give and take, right? So the one that gets oxidized reduces the other one. The one that gets reduced oxidizes the other one. So a reducing agent gets oxidized, an oxidizing agent gets reduced, okay? You might have to watch that again a couple of times, read it a couple of times till it clicks. But the main thing to remember, oxidation is loss, and this guy loses the electron, and reduction is gain. This one gains the electron, okay? So in organic chemistry, those are mostly, we're talking inorganic reactions when we're talking about rusting. But in organic chemistry, redox reactions happen all the time. And a lot of times we don't talk about them in terms of electrons. I mean, there is a transfer of electrons. But with organic compounds, it's often easier to look at whether the presence of oxygen or hydrogen. The loss or gain of oxygen and hydrogen is another way to indicate redox reactions. So this is a nice table that sort of summarizes things. An oxidation reaction is loss of electrons. Oxidation is loss. It also can be a gain of oxygen. That's actually where it got its name from. Oxidation is when you add more oxygen to something. Alternatively, you could lose hydrogen. So with organic compounds, a lot of times oxidation involves either the addition of oxygen or the loss of hydrogen. And then reduction is the opposite. It's either the loss of oxygen or the gain of hydrogen. Okay, so let's look at some examples here. And the examples we'll talk about are with, of course, we'll stick with our monosaccharides. Okay, so here's just a typical aldehyde group that you would see at the end of an aldose monosaccharide. Okay, oxidation of this aldose group involves, check this out, see this oxygen here? We've gained this oxygen here. We've gained oxygen, oxidation. All right, reduction is we either lose oxygen, which we're not losing any oxygen, we still have one oxygen in this molecule, or gain hydrogen. And notice we do have two more hydrogens here. So this one has only one hydrogen group. This one now has 1, 2, 3 hydrogens total. Okay, so aldehydes, aldoses, can either be oxidized or they can be reduced. They can go either way. They can act as an oxidizing agent and get reduced, or they can act as a reducing agent and get oxidized. Okay, when they get oxidized, they become a carboxylic acid functional group. That's a carbonyl with an alcohol. Okay, the whole thing together makes a carboxylic acid. Remember, like we saw with the fatty acids, the ends of fatty acids. And when it gets reduced, it turns into an alcohol. Okay, it becomes an alcohol group. So we can use this nature of monosaccharides to be oxidized or reduced by doing, and we can design tests in order to test for the presence of glucose in bodily fluids, particularly in the urine, which is really helpful for diagnosing diabetes, both in normal, healthy people, but also in my case, it's relevant because it's also used to diagnose gestational diabetes, a type of diabetes that develops when you're pregnant. So prenatal visits usually include a urine test where they test for the presence of glucose. And that test is called the Benedict's test. And it uses this reagent that's blue normally. And if it reacts with Benedict's reagent gets oxidized, okay, so it goes from copper to Cu2O, gains an oxygen. It gets reduced. The copper reagent gets reduced, and I'm not going to bother explaining why. Let's just look at the glucose. Okay, so glucose up here, you have an aldehyde group. After it reacts with this copper reagent, it has an oxygen added to it, so it gets oxidized, okay? This oxidation of glucose results in the reduction of the copper, and that reduced copper turns a red color, okay? So there's strips that are kind of a bluish color, I guess, normally. You dip them in a solution depending on how red they get. Do I have a picture of it? I don't have a picture of it in here, but there's a good one in your book on page 173, actually, showing you the glucose test strips. And depending on how red your urine sample turns the stick, depends on how that is an indicator of how much glucose is in the urine. So the question here is, which reactant gets oxidized? The answer is glucose gets oxidized because it gains this oxygen here. And which is the oxidizing agent? Remember, the oxidizing agent is the one that gets reduced. And the copper gets reduced, so it's the oxidizing agent. It's the agent that oxidizes glucose. Oxidation is lost. It loses electrons to the glucose. Okay? So that is showing how when sugars get oxidized. You can use the ability of sugars to get oxidized to design these laboratory tests to test for sugars. Also, you can do reduction of sugars to form sugar alcohols, and these sugar alcohols are not metabolized as well by the body, and so they actually provide fewer calories. And also, they're not metabolized very well at all by bacteria in the mouth that can cause cavities. So sugar alcohols are excellent sugar substitutes in foods. They're very common low-calorie sweeteners. And they are used in a lot of sugarless chewing gum because they don't result in cavities. And they can be used in other foods to reduce the calories from sugar. Also, the formation of sugar alcohols in diabetics. So diabetics essentially have trouble regulating their blood sugar levels. So they have far too much glucose running around in their blood. And if some of that glucose gets reduced into sugar alcohols, it can cause a type of cataracts in diabetics, these sort of crystal precipitates in their eyes. So there's some relevance there to redox reactions in carbohydrates. So some sample problems to practice oxidation reduction. Okay, determine whether the following substances are undergoing oxidation or reduction. A, iron metal forms rust. Okay, so iron, rust, the chemical formula for rust is Fe2O3, iron oxide. Okay, iron forming rust is an oxidation reaction. We've added oxygen there. Copper 2 ions in solution form solid copper metal on a surface. Copper ions in solution. I'm going to write this down. Hold on, we're on slide 29. So let's write this out. So copper 2 ions, what's their charge? What's their charge? Cu is the chemical formula. And 2 means that it has a +2 charge. Okay? So copper ions have a +2 charge. And solid copper metal is just plain old copper, elemental copper. It has no charge. So which one gains electrons and which one loses? This one has a 2+ charge. This one has a 0 charge. So it has, how do I explain this? If you add 2 electrons to copper 2+, okay, if you add -2, 2 electrons, you get 0. So copper 2+ is the one that's gaining electrons in order to become copper. So gain of electrons, reduction is gain. So this is a reduction reaction. Okay, C, wine containing ethanol, CH3CH2OH, sours to vinegar, CH3COOH. COOH, you may not have seen it written this way, okay? When you write COOH, that is referring to a carboxylic acid group, which looks like this. I'll remind you. COOH. Okay, a carboxylic acid. So, this is an organic compound. These first two were inorganic chemistry examples. So when it comes to organic chemistry, we're looking for a loss or gain of oxygen is a good way, or a loss or gain of hydrogen. In this case, we went from having one oxygen to two oxygens. So we've gained oxygen, which is oxidation. Okay? And then the last one here, the biomolecule NAD plus becomes NADH. Okay, these are molecules that are used in metabolism. NAD plus becomes NADH. This is oxidation is loss or reduction is gain of electrons. This would be a reduction reaction for two reasons. One, because we went from a plus charge to a zero charge, which means we gained negative. We gained an electron, so reduction is gain. Another way to indicate this is reduction is that we gained hydrogens. So reduction can also be a loss of oxygen or a gain of hydrogens. In organic compounds, that's often how we judge. So that table on page 173, table 5.1, that went through those tips about characteristics of oxidation and reduction reactions, that's a good one to star or bookmark or write in your notes. For the take-home test, might actually be smart to make yourself a little cheat sheet of all these things I'm saying that are, helpful for your homework problems. Will make the test probably go faster. Otherwise, just tabbing the page, bookmarking it in your book. Those helpful hints. And practice these homework problems. Okay, so our monosaccharides are sugars in our body. We keep drawing these Fisher projections, right? We draw them as these straight molecules. But they don't actually exist that way in our bodies. They actually exist as ring structures. And the ring structures can actually open up and reform. But the way that they form these ring structures is through basically a reaction where they curl up on themselves and connect into this ring. The reason they do that is because carbonyl groups, the ones in the aldehyde or ketone functional groups and sugars, can react with the alcohol groups, the hydroxyl groups, on another carbon in the sugar. So what happens is, here's your aldehyde group, here's your carbonyl. This is a polar bond, right? Oxygen is an electron hog, so it's going to be slightly negative over here, and this carbon is going to be slightly positive. Whereas this is also a polar bond, and your alcohol, your hydroxyl group, the oxygen is slightly negative. So this oxygen, slightly negative oxygen, is going to be electrostatically attracted to this slightly positive carbon. It's just attraction, opposites attract. And what happens is they end up being attracted and then transferring electrons and forming a bond. So you end up with what's called, a hemiacetal group, okay? This whole thing is called a hemiacetal functional group. You don't really need to know that. I don't care if you know the name hemiacetal or not, but I'm giving you the information just so now you know. What I do want you to know is that when this happens, when monosaccharides form rings, how can I show this picture? Here we go. Okay, so a hemiacetal ring, so this carbon can attack this hydrogen down here, this alcohol group down here. It can attack it from this way, from the bottom, or it can attack it from the top. Okay, it can attack it from one direction or the other. and depending on which direction it attacks it from, it causes that alcohol group that forms to be either on the top or on the bottom. So let's look at this. Okay, so this carbon here, carbon number 1 of the aldehyde group in this glucose molecule, is going to attack this alcohol here down on the 5th carbon. In this picture here is what we're looking at, okay? So the blue oxygen and hydrogen are labeled here, okay? And the first carbon is right here, so they're forming a bond this way. So what ends up happening is you can have this this oxygen group here can either be on the top of this carbon or the bottom. So in this case, the oxygen, or the OH group, the alcohol group, ended up on the bottom of the ring. And when I say on the bottom of the ring, I'm comparing it to if this 6th carbon is facing up. That's sort of the standard way of writing it, with the 6th carbon on the top. So if the 6th carbon's on the top, see carbon number 6. If the 6th carbon is on top of the ring, and this alcohol group off of carbon 1 is on the bottom, okay? Or you could have the alcohol group be on the top, depending which way the attack took place, okay? If that makes any sense. I wish I could demonstrate it better. What if I have, if I just have like a piece of paper here, okay? So you have this straight piece of paper, okay? And you want to make it into a ring, okay? You could do it either this way, fold it forward, or you could do it this way and fold it back. So you imagine that if something's sticking off the side, it could have different consequences based on how this molecule attacked itself, whether it bended forward or it bended backwards, okay? So what this means is that there's 2 different ring arrangements that can happen, and these 2 different ring arrangements are called anomers. It's another sort of, we have isomer, stereoisomer, and enantiomer. Now we have another mer, and these are referred to as anomers. Okay? If that OH group ends up on the bottom, then, and the carbon number 6 is on the top, then you have a trans anomer. Okay? And we call this one the alpha anomer. That's what that little fish-looking symbol means, alpha. Okay? The alpha anomer has the OH group on the bottom. In other words, it is trans to carbon 6. The beta anomer has the OH group on top, so it is cis. It has a cis relationship to this carbon number 6 group up here. Okay? Alpha, these 2 groups are trans. Beta, these 2 groups are cis. So when we're talking about cis and trans with these anomers, we're specifically referring to the relationship of this group, the carbon 6 group up here, and this alcohol group coming off the first carbon. Carbon number 1 here is referred to as the anomeric carbon, because it determines, the groups coming off this carbon determine which anomer it is, alpha or beta. So that's where that terminology comes from. So let's do some practice problems. Okay, let's determine whether the following carbohydrates are alpha or beta monomers. Okay, this should say alpha or beta. So we're always comparing this carbon up here, all right, with the anomeric alcohol group. So here's the anomeric carbon right here, and here's our alcohol group. So are these 2 groups cis or trans? Are they on the same side of the ring or opposite sides? They're on opposite sides, and if they're on opposite sides, we call that relationship trans. Trans meaning across from. All right, so this would be trans, And in terms of anomers, the trans anomer is the alpha anomer, okay? So I'm going to write alpha. That's an alpha. It looks like a fish. Okay, now the second one here. Again, we're always comparing this group to the alcohol group coming off the anomeric carbon. I just noted the anomeric carbon, so here's the alcohol group. Are these on the same side of the ring or opposite sides? They're on the same side, so they have a cis relationship. If they are cis, then it's a beta anomer. Beta anomer. All right, last one here. We've got, this is a pentose, okay? Wait, one, two. No, this is still a hexose because we've got 1, 2, 3, 4, 5, 6 carbons, okay? But now it's a 5-carbon ring. But still, the 6th carbon is hanging up here, and the anomeric carbon here, the alcohol group, is right here. Let me just double check that I am correct. Yep. So it's always the alcohol group that we're comparing to. So it's on the same side of the ring. So it's a cis relationship, which means, again, it's a beta anomer. Beta anomer. Okay? Again, practice those. So when it comes to monosaccharides or simple sugars, we've got those monosaccharides, and we've talked in detail about the different monosaccharides and how to do redox reactions with the monosaccharides, and how the monosaccharides can then flip or sort of chase their tail a little bit and circularize and form a ring structure, okay? And glucose is a very important monosaccharide in our bodies, but a lot of the foods in our diet contain disaccharides. And disaccharides are formed by combining 2 monosaccharides, bonding them together, and that linkage, that bond that forms between them, is called a glycosidic bond, named after the fact that it's between 2 sugar molecules. So a glycosidic bond. So here's an example of alpha D-glucose plus another alpha D-glucose, so 2 alpha glucose molecules connecting to form this glycosidic linkage here. And 2 glucose molecules together form a disaccharide called maltose. Okay? So the way that what we call a reaction, how should I say this? Glycosidic linkages, or glycosidic bonds, are formed through a type of reaction called a condensation reaction, okay? And the opposite of a condensation reaction is a hydrolysis reaction. So in this chapter, we're learning about 2 different types of chemical reactions. Hold on. I guess I owe you guys donuts because my phone keeps going off. So redox reactions, similarly, there are sort of opposite reactions. You have the gain of electrons and the loss of electrons. Condensation and hydrolysis reactions are the gain and loss of water in a reaction. Okay, so you have, in a condensation reaction, you're producing water. Water is lost. Think about having an ice-cold drink on a hot day. You're sitting outside, and what happens? All these water droplets form on the outside of your glass, and that stuff is called condensation. When water condenses from the air onto a cold glass. Okay, so same thing in a reaction. When water is a product of the reaction, when water falls out of the molecule, it's a condensation reaction. Likewise, if you go the other way, you get water is a reactant. If you're going in the reverse direction, water is a reactant, and water gets split into 2, and into 2 pieces that then get added on to the 2 molecules that form as a result of a hydrolysis reaction. So a hydrolysis, the term hydrolysis means to break water, Okay, so condensation reactions, in a condensation reaction, two molecules come together, and as a result, water is formed. So in each of these, it's highlighting where the water is coming from. So this molecule loses a hydrogen, this one loses an alcohol group, and together those two form a water molecule, and you're left with a bond that looks very similar here to this glycosidic linkage. And look, water is a product. So two monosaccharides together is a condensation reaction that results in a disaccharide and water. And then likewise, when there's enzymes in our body that actually have to break down maltose into glucose, we do a hydrolysis reaction where we use water to actually break this bond and end up with two glucose molecules. Okay? So we can practice, some condensation and hydrolysis reactions here. We have, okay, so these are three different problems. Slide 36. Sorry for the cell phone interruptions. My best friend is getting married right now, and I can't be there because I'm too pregnant. So the other bridesmaids are texting me pictures. So this first reaction, we have three different reactions here. In this first reaction, we have this long molecule here. These little, we don't know, let's pretend these are monosaccharides, okay, these circles. So we add water. Water is a reactant, and as a result, we've broken this large molecule up, and we've broken the water up and added it too. So is this a condensation reaction or a hydrolysis reaction? So it would be a hydrolysis reaction because water is a reactant. We break it up, and we use it to break up this larger molecule. So in a hydrolysis reaction, you break down the polysaccharide, okay? How about the second one here? We have two, let's assume these are two organic molecules, okay? And then as the products, we have this one organic molecule. It's all bonded together, and water as a product. When water is a product, it's a condensation reaction, okay? So this is a condensation reaction. These two molecules become bonded together, and as a result, we end up with water. And then lastly here, we have two monosaccharides, two beta-galactose molecules, beta-galactose plus beta-glucose. And they form together, in a product, we've got a disaccharide plus water. So what would this be? Condensation, a condensation reaction. Whenever water is a product, it's a condensation reaction, okay? So that's the thing to look for, really. When you're doing condensation or hydrolysis reactions, look for water. If it's a reactant, if it's on the left side of the equation, it's a reactant, and therefore you have a hydrolysis reaction. If it's on the right side of the equation, it's a product, and that would be a condensation reaction. Okay, so a little bit of nomenclature when it comes to glycosidic bonds, which means we're talking about monosaccharides attached together. We have to name them. We have to indicate which monosaccharides are attached and how they're attached. Okay? So the convention for naming glycosidic bonds is to specify the anomer that's bonded and the number of the carbon that's bonded. So what am I talking about here? So remember in previous slides, here we go. This is a good one. We number the carbons in a sugar, okay? 1, 2, 3, 4, 5, 6. In a Fisher projection this way. In the ring, 1 is the anomeric carbon here, and 6 is the one that's sticking up at the end. So it goes 1, 2, 3, 4, 5, 6. Okay? So when we're naming our glycosidic bonds, we have to know which number carbon. Okay, so in this case, the anomeric carbon 1 is bonded, of the first molecule, is bonded to the fourth carbon, okay, of the second molecule in the bond. 1, 2, 3, 4. So it's a 1 to 4 linkage, is what we call this. And both of these, we've got alpha glucose and alpha glucose bonded together. So this would be an alpha 1,4 glycosidic bond. It's between two alpha molecules, bonding carbons 1 and carbons 4. Okay, so not that difficult, makes total sense, right? This one would be what's called a beta 1,4 glycosidic bond. In this case, we have the beta monomers. Okay, so if you look here, these are the alpha monomers. The OH group is down and the hydrogen group is up. But if you look at these two guys, the OH group, well, here it is, the OH group is up on this one. The hydrogen is down. This used to be the OH group is up. So these are actually two beta anomers of glucose that are bonded together in a beta 1,4 linkage. It's still the one anomer, or the one carbon 1 is bonded to carbon 4 here, are connected through this bond. So this is a beta 1,4 glycosidic bond. And we're going to talk about different polysaccharides, okay, polysaccharides being long chains of glucose that are all connected, and the different types of polysaccharides that form based on the different types of linkages and anomers that you have. So you could have two glucose molecules bonded together, okay, and it could be two totally different compounds is what you end up with. And that's what this is trying to indicate. If you have two alpha anomers of glucose that are bonded together in a 1,4 linkage, you have a disaccharide called maltose. Okay, that's malt sugar that you find in that you use to make beer. If you take two beta anomers of glucose, okay, so just rings that formed by flipping the other way, and you connect them together through a 1,4 linkage, you end up with cellobiose, which is a completely different molecule, which our bodies don't break down for energy, and actually contribute to structural components in plants. So these are two totally different disaccharides, but they're both made up of two glucose molecules, just flipped in opposite directions. So it's kind of crazy how these little tiny details can make a big difference. Little tiny chemistry details can make a big difference biologically in their functions. Okay, so some important disaccharides where you have two monosaccharides connected by a glycosidic linkage. Maltose, which is malt sugar, you find it in malted barley. It's the sugar that's used to make malted beverages, beer, which is made up of two alpha-D glucose molecules. Okay, lactose is milk sugar, which you find in dairy products, and it's a combination of galactose and glucose, specifically beta-galactose and beta-glucose. And sucrose is the, table sugar, and it's a combination of glucose and fructose, specifically alpha-glucose plus beta-fructose. Okay? And these are the different glycosidic bonds. So both maltose and lactose have a 1,4 linkage. But notice here that the linkage between glucose and fructose is a glycosidic bond between carbons 1 of glucose and carbon 2 of fructose. So it's a 1,2 linkage, not a 1,4 linkage. Okay? So like I said, you can connect long chains of glucose together or other monosaccharides in order to make polysaccharides. So polysaccharides are large molecules, and they sort of come in two different categories. They can fall into two different categories. You have storage polysaccharides, which are long chains of glucose that are used to store energy, to store that glucose so that at a later point you can break it down and make it into glucose and then metabolize that glucose for energy, for ATP. And the important types of storage polysaccharides are starches, which you find in plants, and glycogen, which you find in animals. And structural polysaccharides are you can identify them chemically because they only have alpha glucose units. They're all alpha glucose units. Structural polysaccharides, contain only beta glucose units. And that causes them to form in a different pattern. When they form these polysaccharides, the linkages are a different pattern. And these are polysaccharides that actually form structural components of plants, like leaves and stems and roots and stuff like that. And also we'll see chitin, which is a structural component of like insect exoskeletons. Okay, so the polysaccharides that are in so here's some examples of some polysaccharides. You have starches and glycogen. Okay, these are different storage polysaccharides. And fiber is one of these structural polysaccharides. And this is just sort of cartoon drawings showing you the difference in their structures due to the different types of glycosidic linkages they're going to have. So starch is the storage polysaccharide found in plants, and it comes in two forms, amylose and amylopectin. So let's go through this picture again. Amylose is a long, straight chain. Amylopectin is a long chain with branches coming off of it, okay? So amylose is a bunch of glucose units that are bonded in an alpha 1,4 linkage, okay? So just like these maltose molecules. In fact, when you break down, so here's an alpha 1,4 linkage. So long chains of this are amylose, okay? Amylopectin is a bunch of D-glucose units connected by alpha 1,4 linkages. So it's just long chains of amylose. But occasionally there's an alpha 1,6 linkage that forms these branches. So that's how these branches form. So all of these guys in this chain here are alpha 1,4 linkages together. And then at the branching carbons, there's also a 1,6 linkage going on. There's an alpha 1,6 linkage going on that causes that branching structure. So glycogen is the storage form of polysaccharides found in animals, and it's very similar to amylopectin, okay, except that it has more branches, essentially. So amylopectin branches every 25 units, and glycogen branches every 12 units. So you can maybe kind of see that here. This is definitely not drawn to scale, but the glycogen has more branches than the amylopectin does. But same thing, this long chain here is 1,4 linkages, and these branch points have alpha 1,6 linkages. So here's another picture I like, showing you the different polysaccharides. So glycogen is found in muscle and liver cells in animals, whereas starch this is amylose here, is found in plants, the plant that this little guy's eating. And then cellulose is a type of structural polysaccharide that will be on the next slide. It has beta linkages, it's all beta anomers, and it has all beta linkages. So here's the two different types of structural polysaccharides. Cellulose, which makes up the fiber in plant tissues, and it's composed of beta 1,4 linkages. So we've got alpha 1,4 linkages in starch and beta 1,4 linkages in cellulose. So essentially, just the same glucose molecules, except a different anomer, and then they're all attached in a chain, but it causes it to have a different shape and a different structure, and it allows them to be straight. So actually, this is why I like this picture here, because that starch chain, the amylose, even though of all those alpha 1,4 linkages, it causes the molecule to be, it's straight, but it actually curls in like a helix, so like a telephone cord. So, like if you pull it out, it's straight, but it actually coils. So amylose is like this coiled shape due to those 1,4 linkages. That's what happens. In the beta 1,4 linkages, it results in these very actually straight chains. They don't coil, they're straight, which allows them to pack very densely together and form these structural, rigid structures like scaffolding in a building. Another structural polysaccharide is chitin, and chitin makes up the exoskeleton of a lot of insects, that like hard shell, that outer skeleton, I guess. And also it's in crustaceans like crabs and lobsters and crawfish and stuff like that. And it's slightly different. It's not made up of glucose. It's made up of N-acetylglucosamine, which is like a modified glucose molecule. So a whole bunch of those N-acetylglucosamine monomers that are linked together through beta-1,4 glycosidic bonds. So I'll show you. This is a molecule of cellulose from your book, okay, showing you the straight chains of these beta-1,4 linkages, okay, versus chitin. So here's what a molecule of beta-N-acetylglucosamine looks like. It looks kind of like a glucose molecule, right? Except that on this carbon down here, instead of having just an OH group, it has this amine group, this nitrogen, this whole thing right here. Okay, this is called this is the acetylamine. So it looks very much like glucose, and it connects in the same way that it looks a lot like beta glucose, and it connects very much in the same way that beta glucose in cellulose connects. And it similarly forms a very tough type of fibrous material that acts as the exoskeleton of this beetle here. Okay, so here's a long polysaccharide, or it's showing you a portion. That's what the parentheses are saying, that it's just showing you a portion of a long polysaccharide of these beta-acetylglucosamine molecules attached together by carbons 1 to carbon 4, okay, 1,4 linkage. Okay, so the neat thing here, and sort of common theme in biochemistry that you'll see, and also in anatomy physiology that you'll see, is this term here, structure equals function. The structure of a molecule equals the function of that molecule. Same thing with organs and organ systems in your body. The structure of the cell or the structure of the organ is what determines the function of that system. So when it comes to cellulose, you have this straight chain that can then fold up and form this very tightly packed, rigid structure, which gives plants a tight, rigid cell wall, a tight structural support system. So the molecule itself is rigid, and it gives rigidity to the plant. Amylose is these the alpha-1,4 linkages, which actually cause a slight kink in the molecule, which is why that straight chain of alpha-1,4 causes a coiling shape, okay? So it's actually a coil, long coil, okay? That allows, what are coils great for? So you have a telephone cord, that was my example. Telephone cord is maybe 10 feet long. You don't want a 10-foot long string hanging out in your room, so the reason that they're coiled is to save space. It saves space. Same thing with starch in a molecule. This is the storage form of glucose. If you want to store glucose, you're trying to save space, right, in storage. So instead of having this long chain of glucose, it's all coiled up for better storage purposes. Great space-saving structure. And the branch structure of amylopectin and glycogen are adaptive because when it comes to breaking down these molecules to get glucose from them, the breakdown doesn't usually occur in the middle. It usually occurs right at the end. At only the glucoses at the ends of the molecule can get cut off to make glucose, free glucose for making ATP and energy. So the more ends of the molecule you have, in other words, the more branches that it has, the better you can utilize it and break it down quickly for energy. So this structure is specifically designed to make it faster to break down to get glucose from it. So structure equals function of all of these things. The branching structure of glycogen and amylopectin allows for rapid breakdown and utilization of glucose. The long coiled structure of amylose allows for maximum storage potential, so compression. And the flat, the tightly packed, rigid structure of cellulose gives the quality of rigidity to the plant tissues. So, sample problem. Identify the polysaccharide described by each of the following. A polysaccharide that is stored in the liver and muscle tissues. That would be, ding, 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 glycogen. Glycogen is the polysaccharide that is stored in liver and muscle tissue. That's the definition. B, a component of starch containing only alpha-1,4-glycosidic bonds. There's two components of starch, remember, amylose and amylopectin. So which one has only alpha-1,4-glycosidic bonds? That would be amylose, the long one that's actually coiled up. And C, a polysaccharide found in the exoskeleton of insects. That one is chitin, chitin, C-H-I-T-I-N, pronounced chitin. All right, so the last part of the chapter talks about oligosaccharides. So monosaccharides are like sugars, simple sugars in our food, like glucose and fructose. Disaccharides, two monosaccharides bonded together, also common sugars in our food, like sucrose, table sugar, lactose, milk sugar. And then we talked about polysaccharides, which are the fiber in our diet, in terms of, well, it can be the fiber. Also, so the polysaccharides are the complex carbohydrates. Your starches, amylose and amylopectin, and fiber. And then also in our bodies we have glycogen. So those are all the polysaccharides. Then we sort of skipped over the oligosaccharides because oligosaccharides aren't really components of our food. We break down polysaccharides into oligosaccharides, briefly as we're cutting them into ultimately monosaccharides. But oligosaccharides are actually used in our body as what I like to refer to them as cellular name tags, okay? So you have a cell, and it has a little name tag that says, hello, my name is liver cell, my name is, nerve cell, my name is kidney cell, okay? All of the cells in our body literally have tags on them, oligosaccharide tags, that tell other cells who they are, where they're from, what they do. Okay, so the cell membrane is composed of lipids and proteins, fats and proteins. And these sugar groups, these oligosaccharides, can attach either to the lipid or to a protein to form these little surface tags. So, I don't have a good picture of it, but so you just kind of have to imagine it in your head. So little oligosaccharides sticking off the cell. So one example of a group of oligosaccharides that act as name tags, cellular name tags, are the A, B, and O blood antigens. Okay, they are all oligosaccharides. So you have three different blood types, O, A, and B. So type O blood has this oligosaccharide attached to it, okay? Type A has this oligosaccharide. It's 3 or 4 monosaccharides bonded together, so it's only a few saccharides, so therefore an oligosaccharide. And then type B oligosaccharide looks like this. You don't have to memorize these or anything. But what you do need to understand is that the body makes antibodies towards this extra group, I guess, that sticks off the oligosaccharide. So type O doesn't have anything sticking off of it. So there are no antibodies that are made towards type O blood. But your body does make antibodies to type A and type B. But not necessarily, I shouldn't say that. If you have type A blood, your body does not make antibodies to your own blood. It's fine with that. But it does make antibodies towards type B blood. It'll make antibodies to this extra galactose group here. And therefore, if you give someone with type A blood, type B here, I should just move to this picture. It's a better example, okay? If you have type A blood, you have type A antigens, this type A oligosaccharide. Type B blood, you have B antigens. If you have AB blood, it means your blood cells express both A and B antigens. And if you have type O blood, Your blood cells express O antigens, which are essentially invisible to the immune system. Okay? So if you have type A blood, it means you make type B antibodies. If you have type B blood, you make type A antibodies. If you have both A and B in your blood, you don't make any antibodies. If you have type O blood, you make both A and B antibodies. So what does that mean? Whatever you make antibodies to, that means your body will attack that antigen if it sees it. So if you have type A blood, and you get blood from someone who has type B, or A and B, those antibodies, those anti-B antibodies, will attack those blood cells. So you don't want to give someone with type A blood any kind of blood that has B antigens in it, because that person's body has all these B antibodies that will attack it. So O, on the other hand, okay, well, let's go with A. Let's do this first. AB, people who make A and B antigens, don't make any antibodies. So you can actually give them any blood type, A or B or O, and they will accept that blood without attacking it. So that's why AB people are the universal acceptors, okay? Positive and negative, when you say AB positive or AB negative, the positive and negative actually refers to another antigen group called the Rh antigen. So if you're positive for everything, if you're positive for A and B and Rh, okay, it means you don't make any antibodies. So your body is very accepting of foreign blood cells. If you have type O blood, your body is very picky about what blood cells it will take, because it won't take A or B because it will attack both of them because of those antibodies. So people who have type O blood can only receive blood from other people with type O blood. So they are particularly, type O blood is particularly wanted by blood banks for that reason. The other reason that type O blood is very valuable is because O is a universal donor, because the O oligosaccharide, that O antigen, is essentially invisible to the immune system. Notice, none of these types make anti-O antibodies. There's no such thing. O oligosaccharide is invisible to the immune system. So you can give O blood to any of these people, and they will be fine. They will not attack the O. So O is particularly valuable blood in a blood bank, particularly O negative, because that means there's no O group, there's no there's an O antigen, which is invisible, and there's no Rh group, the other type of blood group. Okay, so the last slide here is a sample problem about blood donation. Explain whether the following blood types could be donated to a person with type A blood. Type A blood, okay? So, a person here, I'm going to do this actually, temporarily. Why not? Alright, it won't let me paste here. Whatever. Okay, type A blood. Someone with type A blood, could they receive type A blood? Yes, they can. Someone with type A blood, can they receive type B blood? No, they can't, because someone with type A blood makes anti-B antibodies. They would attack it. What about AB blood? Could they receive AB blood? No, they can't, because there's still B antigen on those blood cells. Still has B antigen there, so someone with type A blood would still attack these cells because they have B antigen on them. What about type O blood? Yes, they could accept type O blood. Anyone can accept type O blood because there's no anti-O antibodies. Okay? So hopefully that lecture is clear about the different types of carbohydrates, the different classes of carbohydrates, the structure of carbohydrates, how they form these ring structures, and how then those ring structures can combine through glycosidic linkages to form longer chain, either to form disaccharides or to form polysaccharides. And we didn't, in this chapter, we don't really discuss the role of carbohydrates in making energy. We save that for the chapter on metabolism. But we do talk about oligosaccharides and how important they are for our cells to recognize one another in terms of blood groups. And the two key reactions we talked about were redox reactions, oxidation reduction reactions, and condensation hydrolysis reactions, okay? So I know it's kind of a lot going through one chapter in a whole lecture. It takes nearly 2 hours, which is usually more like 2 class periods, okay? So if you need to rewatch this lecture or watch it again after doing some of the homework problems, that's fine. But make sure you're reading through the chapter and working the homework problems, okay? And that's all. Thanks for watching. Bye.